There we go. So our participant numbers are slowly rising. There we go, 14, 16.
Wow.
24. We'll just give them a few minutes for the numbers to settle, and then we'll get cracking.
Yeah, for sure.
Up to 50, so we're halfway there.
Wow.
Okay, I think if there's anyone else who wants to join, we'll let them come in their own time. We'll make a start. So, good evening, everyone. My name is Puru Thambiayah, and I will be hosting this webinar this evening. So I'd like to start by addressing the interesting surroundings we find ourselves in. I'm pleased to see so many freshers who've signed up to this event. And while this may not be the university experience you originally anticipated when you submitted your UCAS applications in October 2019, this should not hinder you from making the numerous special connections university life provides you with before you enter your medical careers. Whilst we interact in a virtual world now, there will come a day where we will meet in person again. And no doubt Professor Pepper will return in the flesh for this very special reunion. So today we have an exciting webinar delivered to you by Professor Chris Pepper, who is the Chair of Cancer Research at BSMS. Older years will remember with fond nostalgia, and freshers, you will look forward to this. Professor Pepper introduces the cancer teaching in Module 102 and delivers an entertaining CLL symposium alongside his lovely wife, Dr. Andrea Pepper, and his colleague, Dr. Tim Chevasu. Professor Pepper's career in research spans over 30 years. He completed his PhD in medicinal chemistry at the Welsh School of Pharmacy in 1992. In 2005, he was appointed senior lecturer at the University of Cardiff, focusing on leukemia research. And subsequently, he was appointed reader in hematology and then professor in 2013. He was also appointed Dean of Research at Cardiff University in 2014, a role he held for 3 years before arriving at BSMS in 2017. Professor Pepper is unique in that he has focused on CLL during the majority of his career, and his work is almost entirely patient-focused. He has over 240 publications with almost 5,000 citations, so he is definitely an academic heavyweight. But more importantly, he is an inspirational speaker and a passionate educator. And we are incredibly fortunate to have him present here today to deliver a rather special webinar tonight. So, without further ado, I will hand over to you, Professor Pepper.
Thank you, Puru. That's very, very kind. Hang on, let me just share my screen. Oh, that's a good start. Screen has failed to share. Hang on, let me try again.
That's perfect.
Okay. How does that look?
Wonderful. Over to you.
Excellent. Thank you, Puru. Okay. Well, first things first, academic heavyweight. Well, I'm trying to lose a bit of weight, actually, and lockdown really helped me to do that. But nonetheless, I thank you for your very kind words, Puru. And also thank you to you and your OHP organising committee for inviting me to speak at this first event in your calendar for 2020, 2021. It's a real pleasure for me to come and speak to you this evening, especially as it's about my favourite subject, which you rightly identified as CLL, Puru. So I really hope that everybody that's dialed in this evening is going to think that it's worthwhile, and hopefully you'll enjoy the next sort of 35, 40 minutes of me taking you on a whistle-stop tour of CLL. I mean, Puru didn't mention this at the start, but really would like you to engage with me after the talk and hopefully have some interesting questions that you want me to try and wrestle with. I'll do my best. But without further ado, let's get cracking. So the title of my talk is Biological Insights and Clinical Advances in CLL. And I guess the most pertinent part of this title is the personal perspective. So, I mean, this is not a disclaimer, but I would like to just say to you that everything I'm about to say to you is really based on my own personal views. But like a good scientist that I hope I am, most of it, if not all of it, will be evidence-based. Okay. So the outline of my talk, what I'm hoping to try and achieve today is to talk a little bit about how therapy for CLL has evolved over the last sort of 40 or 50 years, and hopefully try to explain to you how the acceleration in the advancement of the treatment of CLL has really been promoted by biology, by trying to understand what makes the tumour cell behave in the way that it does, and then develop smart therapies to try and attack that specifically within the cancer cell. And that has a number of, possible advantages for the patient particularly, because by targeting something that's peculiar to the cancer cell, hopefully we reduce the side effect burden that patients feel. And as most of you will know, most of our standard chemotherapeutic approaches, which still are the mainstay of the way that we treat cancer patients today, have significant toxicity attached to them. So targeting the Achilles heel, if you like, of the tumour cell may well spare normal cells. And that's something that we really ought to be aiming to achieve. Okay, so here's my little timeline that I've put together for you, which basically starts in 1949, a time in history where not even I was alive. But essentially it tracks the introduction of, well, steroids and then chemotherapy. So alkylating agents like chlorambucil and cyclophosphamide, and then these purine nucleoside analogues like fludarabine. And then I'm going to focus most of my talk in what I consider to be the more interesting part of the evolution of therapy, the era of targeted therapies, which was really sort of ushered in by the introduction of monoclonal antibodies, in this case, rituximab. Okay, so as Puru quite rightly said, CLL is my thing. Chronic lymphocytic leukemia is the most common leukemia in adults, certainly in the Western world. And I think it's the most important leukemia, not just because of its prevalence and incidence, but because it's such an interesting pathology that has so much to offer a researcher, both clinical and non-clinical. And we'll talk a little bit about why I think that's the case as we progress through this next 35 or 40 minutes. So the diagnosis of CLL is pretty straightforward. So with the advent of monoclonal antibodies, and that's for this purpose, I mean diagnostic rather than therapeutic antibodies, and immunophenotyping, we can now identify these particular markers on the surface of the cancer cells, CD19, CD5, and CD23, and that's pretty much a bellwether indicator that a patient has got CLL. So the co-expression of CD5 and CD19 in expanded population of lymphocytes, so lymphocytosis, is pretty indicative of a diagnosis of CLL. And what you can see on the right-hand side here, and I've already made a mistake because I haven't got my laser pen set up properly, but here we go. Okay, so what you can see on the right-hand side here is the incidence of CLL with increasing age. So when I first started working on CLL, 25 years ago plus now, CLL was regarded as a disease of the elderly. Now, you as students might still think, looking at this profile, that CLL is a disease of the elderly, but I would beg to differ. I don't feel old, and yet the incidence of CLL at people with my age is starting to rise significantly by the time you get into your early to mid 50s. Okay, so in this case, if you're a male and you're, say, 54 years of age, your incidence of CLL is about 16 per 100,000 of the population. So this is a pretty common disease for men. And it is worth noting here that more men get CLL than women. And we really don't know why. Maybe if we have time and the OHP committee have the inclination, I'll come back and we can discuss why there is an excess of cancers in men compared to women if you exclude all of the hormone-driven tumours. But that's for another day, perhaps. But what I want you to take away today is that the incidence of CLL probably peaks in the mid-70s and then drops away a little bit with every succeeding decade. But remember that fewer and fewer people actually make it into those further decades. So the incidence is still really high. So what we understand about CLL now is that, you know, it's a microenvironment driven disease. And particularly signaling through the B cell receptor appears to be very, very important. So if you like, if you think about normal B cell biology, normal B cells will readily signal through their B cell receptor when they experience an antigen, this so-called antigenic drive. And we think that CLL is simply doing what a normal B cell would do and reacting to an antigen that it recognizes. The type of antigen is still controversial, and we'll touch on that a little bit more in a few slides' time. One of the things about CLL that we still do not know, though, is what causes CLL. What is the pathogenesis of this disease? We know, and I've contributed to the literature on this, that there are familial risk factors. So you inherit certain single nucleotide polymorphisms from your parents that may predispose you to developing a B cell malignancy. But even if you add all of those alleles, risk alleles up, it probably only represents a 5% increased risk compared to somebody that doesn't have any of those alleles. So the genetic predisposition is actually relatively small. Okay, we know, like in most cancers, that genomic aberrations and genetic mutations really matter. They drive the pathology. So the type of genetic alterations that you accumulate in a tumor cell has a huge bearing on how that disease will behave in the future. And actually, we could have spent the whole of this session just talking about the evolution in the way that we characterize genetic mutations in human cancers. But we're not going to do that today. But on the right-hand side at the bottom here is one of these Circos plots, which basically is a way of representing the new way that we identify genetic lesions in human cancers. And this is through next generation sequencing, which is essentially just massively parallel PCR reactions, polymerase chain reactions. Okay, we're not going to touch on this later in this talk at all, but the epigenetics of CLL is also fascinating. So if you like, the imprinting on the human genome that actually drives the expression of genes is different in CLL patients compared to normal individuals. And within that CLL population, we can identify different subgroups based on their epigenetic signature. So how much of their genome is methylated really does have a massive bearing on how the disease behaves. Okay, so why am I interested in CLL at all? Well, this is where the personal bit kicks in. So for the eagle-eyed amongst you, this is the beautiful Professor Chris Pepper. It is very natty bathers. Yeah, I wasn't really a handsome young man, and I turned into an even less handsome older man. But this guy here is the reason why I'm interested in CLL. And he actually was far more handsome than I'll ever be. This was my dad. So, yeah, here he is on the right-hand side. This is the last photo I have of him. So my dad was diagnosed in 1992, the year I graduated with my PhD. And I think it's fair to say that for most of us, you know, our parents have a huge bearing on our lives. They have a massive, usually very positive impact in our lives. But I think I can make a claim that my dad has perhaps had more of an influence on my life than most parents have in their children, particularly in determining their career, because I changed my career based on my dad's diagnosis of CLL. I trained as a breast cancer researcher, really. But when I went to the library in 1992 and looked up this leukemia, chronic lymphocytic leukemia, I was absolutely shocked how little we knew about this illness. And it may have been the impetuosity of youth, may have been a bit of arrogance, or maybe just showing that I had no clue whatsoever what I was getting myself into, but I decided that I would really like to try to make a difference in this disease and try to understand it better. Partly because of a familial interest, and partly also just because I couldn't believe that nobody was researching it, or that's how it appeared to be. Okay. So, back in the 1990s when Dad was diagnosed, there really were essentially just three options therapeutically. So we had steroids, prednisone, we had chlorambucil, or as its trade name here is leukeran. And chlorambucil is an alkylating agent, so it binds into DNA, it causes DNA damage, DNA strand breaks, which results in apoptosis. And fludarabine. So fludarabine is a purine nucleoside analog. Oh, hand up, was that Puru? I can't hear you.
Not at all. Do go on, please.
Oh, sorry, I thought you had your hand up. I thought I'd done something terribly wrong.
No.
Okay, that's a relief all round. Okay, so yeah, fludarabine is a purine nucleoside analog. So essentially what fludarabine and drugs of this class do is they, instead of, during DNA replication, instead of incorporating a normal purine base, fludarabine is incorporated instead. It then cross-links into the DNA strand and causes replication fork stalling. So essentially, both of these drugs, chlorambucil and fludarabine, are DNA damaging agents. They do it in a slightly different way, but they essentially induce toxicity through that mechanism. What I think you can see here is that, you know, if you treat patients with fludarabine or chlorambucil, there was a marginal improvement in progression-free survival if you got fludarabine monotherapy. But what I would really like you to focus on, and we're not going to look at too many Kaplan-Meier curves in this talk, but the Kaplan-Meier curve is very steeply going down for both drugs. So if you like, the median progression-free survival, even for fludarabine, is about 2 years. So these therapies were less than optimal, that is for sure. And then if we look at their effect on overall survival, the Kaplan-Meier curve on the right-hand side here, you can see that Fludara, chlorambucil, and the combination of fludarabine and chlorambucil really were not different from one another. And, you know, the Kaplan-Meier curves, again, were heading south at an alarming rate. So in other words, these drugs really had very limited effect in changing the normal pathology of the disease. And in fact, up until, well, we'll talk about this in a few minutes, but at this point in history, none of the available treatments showed any improvement in overall survival. And it's worth just reflecting on that for a second. What I mean by that is, if you left the patient untreated, they would do as well, if not better, than if you gave them the therapy. So, you know, the first rule of being a good doctor is do no harm. So the question really came about, would spring to mind here, is it ethical even to give a drug like chlorambucil or fludarabine, knowing that it's not going to alter the pathology of the disease? So I'll leave that one hanging, and we can perhaps debate it a little bit later. But the reality is that treatment was reserved until the patient got so poorly that they absolutely had to do something to try to address the tumour. Okay. Okay. So in terms of evolution of therapy, the next sort of drug on the block, if you like, was cyclophosphamide, another alkylating agent. And this is the CLL4 trial, which I was involved in. And I've still been working on these CLL4 samples only recently. So clinical trials produce a repository of cellular material and clinical data that we can harvest for generations after the trial has been done. So there is a real rationale for why clinical trials should be engaged in. But what I want you to note here is that even though the combination of fludarabine and cyclophosphamide appeared to significantly improve progression-free survival, yet again, that combination had no positive impact on the overall survival of patients with CLL. And this was published in 2007 by a collaborator of mine, Daniel Kotowski. So it took till 2009 to confirm that any treatment showed an improvement in overall survival for CLL. And this came in the form of the German CLL8 study. The Germans do clinical trials arguably better than anybody else. And I think it's because they are sticklers for the rules. The German trials are so well designed and so well controlled, they usually yield incredibly good results. They are meticulous. But what this trial showed for the first time is that if you combine fludarabine and cyclophosphamide with the first of our targeted therapies, rituximab, in this case the R, so this is a monoclonal antibody targeting CD20 on the surface of the tumour B cell. They showed an increased overall survival in patients treated with that combination. So this was big news. And actually, to be honest with you, it still is big news for a lot of patients who receive therapy today. So this is why I think I want you to sort of go with me on this journey and focus really on this era of targeted therapies, because the introduction of that monoclonal antibody to that FC combination really did change the game. And actually, it's very interesting to note that R on its own has very little impact on CLL patients at all. So it's the combination of those DNA damaging drugs with a targeted therapy that really brought about the change. So over those almost 2 decades, we saw incremental improvements in survival. But I think it's fair to say that if we were judging clinical trials at that point, the empirical nature of clinical trials produced slow incremental advances in patient outcomes. So the question really, and I hope it's slightly rhetorical, is can we exploit knowledge about the biology of the tumour to improve clinical outcomes? And within that question, can we identify patients who would particularly benefit from different types of treatment? So if you like, the fledgling idea of precision or personalised medicine. And can we identify something about the biology of the disease that would allow us to preferentially target that in the tumour cell? And the rest of my talk this evening is really going to focus on both of those aspects. Sadly, none of the changes in new therapies that we've seen since 2008, have had an impact on my dad, because he died of infectious complications caused by his CLL in 1997. So very poignantly, I carry on researching CLL, knowing that the disease killed my dad. But it's a real motivator for me. And I feel very privileged to work in this area of research. And I'm really grateful that my dad provided me with the sort of impetus to start researching in this field. Okay, coughing away here. Okay, it's not COVID, I'm sure. Okay, so the first question that I want to talk to you about today about the biology of CLL is CLL one disease or two? Now, actually, my COVID aside is quite pertinent here, because this speaks to the normal B-cell biology. So mounting an effective adaptive immune response is part and parcel of what B-cells do. And they work in conjunction with other parts of the adaptive immune system, namely T cells, to refine the immunological response that we make to pathogen. So this really is a very sort of relevant sort of aside in relation to COVID, because when I wear my other hat at BSMS, I teach adaptive immunity. And so what we've got in CLL is apparently all of the B cells are bearing markers that would tell us that they are antigen experienced. But one fraction of CLL patients undergo a process called somatic hypermutation. So their immunoglobulin genes are rearranged in response to antigen engagement. So this is a process that normal B cells undergo to try to develop a higher affinity B cell receptor on their surface. Now, what's really interesting here is that the patients that bear the hallmarks of somatic hypermutation, i.e. they have mutated CLL, they have generally got a much better prognosis than the patients that are still bearing the markers of antigen experience, but have not undergone somatic hypermutation. Now, it's still unclear as to why we have this bifurcation in CLL, but it's likely that what we're seeing here is a different antigen stimulating both tumour cells. So in the case of unmutated CLL, the antigen is likely to be an autoantigen or an antigen, a bacterial antigen, where we can form multimers of that antigen on the surface of the B cell, and the cell does not undergo class switch recombination or somatic hypermutation. It simply produces immunoglobulin in response to binding of the antigen. In the mutated case, the cells are at least trying to react to antigen. But what's really interesting is that they don't undergo class switch recombination. They undergo somatic hypermutation, but they don't produce class switched immunoglobulin in the majority of cases. We get the odd case in CLL where you have an IgG or an IgA immunoglobulin secreting B cell, but most of them secrete what you might term as naive B cell immunoglobulin, or IgM. Okay. So why am I banging on about the biology of these B cells and whether they undergo SHM or not? Okay. Well, the reason why it's a big deal is that whether they're unmutated or mutated has a massive bearing on the natural pathology of the disease. So if you've got unmutated immunoglobulin genes, you tend to have a whole raft of other poor prognostic markers on the surface of the B cell, and those B cells appear to be more proliferative and therefore cause the patient more problems quicker. The mutated group seem to have a more indolent form of disease, and this may be because they become rather anergic. They see the antigen that they recognize quite frequently, but they become tolerized to it. They tend to not react in a proliferative way when their B cell receptor is engaged. So patients with different B cell receptors respond differently to our gold standard, if you like, chemoimmunotherapy. So this is why it's become even more relevant that we bifurcate our CLL patients into these unmutated and mutated subgroups, because the unmutated group does much worse than the mutated group when we treat them with FCR. Okay, and this is data that actually we've only just published, taken from the UK Arctic and Admire trials. So this is very, very up-to-date data. Increases in nursing care and supportive care for patients are very evident now. So cancer patients in general, but CLL patients in particular, tend to survive a lot longer now than they did perhaps 20 years ago, because we're just better at managing their symptoms. And as I said to you about my dad, he succumbed to an infection and had died of peritonitis, essentially. And that was a very, very common cause of CLL death, but not so much anymore because of the way we manage CLL patients. So we've got these mutated and unmutated groups of patients. And this is the only slide where I'm going to talk about differential prognostic markers, because I literally could talk for the whole hour just about prognostic markers in CLL. But what I want you to take away from here is that not only can we identify good and poor responder groups based on immunoglobulin genes, but we can also refine that. So if we look at telomere length in the tumour cell and the expression of this integrin on the surface of the tumour B cell called CD49D, what I want you to take away is that there's a subgroup of CLL patients with mutated immunoglobulin genes, long telomeres, and low expression of CD49D that look like they have an incredibly prolonged survival or response to FCR. And in fact, this is with nearly 10 years worth of follow-up, it's possible that this group of patients, at least 75% of them, might be cured of their CLL using this chemoimmunotherapy. So it really matters, doesn't it, that we identify what type of CLL you have, because surely that would determine what drugs we give you. So again, this reflects back on what I said earlier. We're on the very cusp now, I think, of developing personalized medicine strategies for cancer patients. And CLL is likely to be in the vanguard of this because we have so many great drugs that are currently available to treat this disease. So we've identified the subgroup of patients that I've just told you might even be cured with FCR, but what about the patients with unmutated B-cell receptors? They do badly when we give them FCR. What's available for them? Okay, so what we know about the biology of CLL is that the unmutated cases signal better through their B-cell receptor. And that's why, if you remember back a couple of slides ago, they proliferate quite a lot because their B-cell receptor gets engaged and they react like a normal B-cell would and undergo proliferative drive in response to antigen. So is it possible then that by targeting the signaling through the B-cell receptor, we might be able to affect some positive change for patients with unmutated CLL? And this is a moot point because we now have a number of really interesting drugs that target downstream signaling of the B-cell receptor. The most well-known and best clinically trialed one of these is a drug called ibrutinib. So this targets this kinase here, downstream of the B-cell receptor, called Bruton's tyrosine kinase, BTK. And I think it's not overstating it to say that the introduction of ibrutinib for the treatment of CLL has transformed the world of CLL. It really has. FCR has incrementally changed the game for some CLL patients, but ibrutinib really has been a game changer. Anecdotally, I have a friend who I think has been, well, he's certainly in very long-term remission following treatment with this PI3 kinase delta inhibitor called idelalisib. But idela is not a drug without side effect, and so ibrutinib arguably has a milder side effect profile, so more patients will get treated with a BTK inhibitor than a PI3 kinase delta inhibitor as things stand. So does it make the unmutated cases more susceptible to BCR signaling inhibitors because they rely on that B-cell receptor signal to keep them proliferating and alive? Well, here's some data that Andrew and I published in Leukemia a few years ago, which I think will illustrate the answer. So if we take CLL patient samples from different types of CLL patient, so in this case, they're phenotypically different. So CXCR4 high, CD49D high patients. Now remember, the eagle-eyed amongst you will think back to that unmutated slide, poor prognosis slide. These patients with unmutated genes usually have high levels of 49D, and they often express high levels of CXCR4. So we're talking about the same group of patients here. These are likely to be unmutated cases. And these ones here with the CD49D low, CXCR4 low expression are likely to be our mutated cases in large measure. So what we can see here is that the poor prognosis phenotype samples are in fact more susceptible to killing with ibrutinib and idelalisib than their CXCR4 low, CD49D low counterparts. And very pertinently, and we'll talk about this a little bit more in a few more slides, they're also more susceptible to having their potential to migrate inhibited. We'll talk about migration in a little while. So, does that translate through? So this is all in vitro stuff. This is stuff done in a test tube in the lab. Does that translate through into clinical practice? So when we treat unmutated cases with ibrutinib, do they respond particularly well? Well, I guess I wouldn't be saying that if the answer wasn't yes. And here's the evidence. So this is from the RESONATE trial, one of the largest trials ever done in CLL with ibrutinib. And what I think you can see on the right-hand side, so on the left-hand side are all the patients lumped together. And on the right-hand side, this is patients treated that are unmutated with ibrutinib compared to FCR, fludarabine, cyclophosphamide, rituximab. And what I want you to take away from this is that nearly 91% of the patients are progression-free at 3 years with ibrutinib. Bear in mind, these are the patients with the most aggressive disease that are likely to succumb to their disease very quickly without therapy. So this is a remarkable result. So it appears that the unmutated CLL patients show a durable response to ibrutinib and definitely show superior responses to those if they were treated with FCR. So you can see already that we now have potentially 2 different therapies for 2 different groups of CLL patients. So we're going to just transit into or traffic into CLL migration, if you'll excuse the pun. So CLL essentially exists in 3 microenvironments in any given patient. So there's the obvious vascular niche, which gives the disease its name, because leukemia means blood cancer. Yeah. So if it's not in your blood, it's not a leukemia. Yeah. But CLL is actually a lymphoma. So CLL is a hybrid disease, if you like. So it originates in the lymphoid tissue, but lives at least part of its life in the peripheral blood. And what we now appreciate that we didn't know before is that there's constant trafficking of these CLL lymphocytes between the 3 microenvironments in which they live. So the bone marrow niche is predominantly a pro-survival niche, and the lymphoid niche is where all the action takes place. So all the proliferation that we see in CLL takes place in lymphoid tissue, in the lymph nodes of CLL patients. So it stands to reason, therefore, doesn't it, that the trafficking of CLL lymphocytes really does matter. So if we can block the egress and or ingress of CLL lymphocytes into lymph nodes, we potentially can switch off the proliferative drive of the tumour. And indeed, that's been shown to be the case from the clinical effects that we see when we use BTK and PI3 kinase delta inhibitors in the clinic. So on the right-hand side here, what we've got is this sawtooth profile of lymphocytosis that we see with CLL patients treated with ibrutinib. Now, it's saw-toothed in this diagram because the treatment was pulsed. So the patients received the drug. It was sort of a 2 weeks on, 2 weeks off protocol. So every time the patient went onto the drug, their lymphocytosis in their peripheral blood spiked upwards. Now, if you can imagine what the doctor that gave the first dose of ibrutinib must have felt when they noted that their patient's peripheral blood lymphocyte count had rocketed through the roof when they gave this wonder drug, ibrutinib, you can imagine it was a bit of a squeaky bum moment. Because, as I said to you at the start, part of the Hippocratic Oath is do no harm. So this is potentially an indication that the disease is being made worse by the therapy, not better. Fortunately for those treating doctors, that wasn't the case. What we worked out was, and the yellow line here shows it really nicely, this is the average lymph node size or lymph node involvement of the disease. So when those spikes in lymphocytosis were being observed when we treat with the drug, the tumour is actually being ejected from the lymphoid tissue, and that's what's causing the increase in lymphocytosis. So we're moving the CLL lymphocytes out of the lymphoid tissue where they're proliferating into the peripheral vasculature where they are not proliferating. Okay? And what we've worked out now is that if we keep giving ibrutinib for long enough, those lymphocytes eventually start to die off and their lymphocytosis resolves, as you can see out here at, well, a year and a half out from treatment. So it's a fascinating class effect and all the Bruton's tyrosine kinase inhibitors that are currently in the market do this, but fascinatingly, so do PI3 kinase delta inhibitors. So this is not an on-target effect of BTK inhibition per se. This is probably a consequence of switching off some downstream effect of signaling through the B-cell receptor. And yeah, like I said to you earlier, if you can stomach it, Puru, you can get me back and I'll talk to you about NF-kappa B in the future, my favorite transcription factor, and how this might be being modulated by ibrutinib and PI3 kinase delta. Okay, the key here is to note that ibrutinib, even though it is a wonder drug and most patients tolerate it, it is not curative. Now, why is it not curative? Well, one of the reasons why ibrutinib is not curative is that although it's very good at getting quite a lot of the CLL lymphocytes out of the peripheral blood, out of the lymph node into the peripheral blood, not all of the tumour cells leave the lymphoid tissue. And so you have residual disease that can come back to bite the patient. So the question we really need to address now is, how can we treat this residual disease? And this actually, fortuitously for me, for the narrative of this story, takes me back to the start of my postdoctoral life as a CLL researcher. So in 1995, I started working on CLL. And the first thing I worked on in CLL was a family of proteins called BCL2 family. The reason why I was so interested in this disease and so interested in BCL2 is that CLL lymphocytes hang around for a long time. So it was always considered that CLL was a disease of failed apoptosis rather than increased proliferation. We understand now, 25 years on, that actually that was a gross oversimplification of the truth. And actually CLL is a very proliferative disease. But it still holds true that CLL lymphocytes are reluctant to die. And part of the reason why they're reluctant to die is the overexpression of BCL2 protein. Okay? So BCL2 was described actually in 1990 by a guy called Stanley Korsmeyer, and it literally was the first in a new class of proto-oncogene that really defined, you know, what an oncogene really meant. Because up until that point, virtually all oncogenes conferred a proliferative drive or a proliferative advantage to cancers. BCL2 did not do that. It actually put the brake on cellular apoptosis. And yeah, and I published my first paper on this subject in 1996. It was the first paper to be published on BCL2 in CLL. And actually we went on to demonstrate that if you inhibit BCL2, in this case using antisense oligonucleotides, that you could actually reset the sensitivity clock, if you like, within the cell and make the cells more susceptible to apoptotic cell death. So, you know, if you like, this was the forerunner or the proof of concept that targeting BCL2 as a therapy, as a therapeutic strategy, could work. So we're 20 years later, and we are now targeting BCL2 with this drug called venetoclax. Now, venetoclax, it works, it's a BCL2 inhibitor, but what it does is actually displaces these so-called BH3 only proteins, which makes the cells undergo apoptotic cell death. So it's a very, very clever drug. It's a BCL2 mimetic drug. So it basically tries to mimic the structure, the three-dimensional structure of BCL2. Okay, so does it work? Well, yes, it does. You can see very clearly here that when we use venetoclax, in this case, in combination with that first of all of our targeted inhibitors, rituximab, we see really positive signs that this drug is having a significant clinical effect. But nobody's been cured with venetoclax either. So we are now trying to combine, perhaps, and I hope slightly less empirically, ibrutinib with venetoclax. And I think this slide illustrates the point that combining two drugs with two different mechanisms of action can really have a significant synergistic effect. So here's, on the left-hand side of this plot, in the blue bar, you can see the effect of ibrutinib as a monotherapy after 3 cycles. So the vast majority of patients are showing a partial remission. Yeah. So for the purposes of this discussion, a partial remission is a 50% reduction in tumour burden. Only about 1% of the patients show a complete response, right? And none of them show a molecular remission. In other words, they can still detect disease. But if we then add venetoclax to the soup, you can see in the orange bar an incremental advance or an incremental increase in undetectable minimal residual disease. So by the time you get out to 18 cycles of venetoclax combined with ibrutinib, 69% of the patients are showing MRD negativity. So that's really interesting, isn't it? And potentially very exciting that these patients, some of these patients at least, might be cured. Now, it's too early for us to say for sure whether ibrutinib and venetoclax is going to cure these patients, but nonetheless, I hope you'd agree with me that it looks like we're on the right track. But, and here's the rub, cancer cells are tricky customers. So, This paper was published only a couple of years ago now, which demonstrated that the combination of using venetoclax and ibrutinib actually caused an activation of non-canonical NF-kappa B signaling via toll-like receptor 9 activation. So, perhaps not surprisingly, this is the focus of our current research at BSMS. So Andrea and I are now trying to understand how toll-like receptor 9 signaling is induced as a resistance mechanism to venetoclax and ibrutinib. And what's intriguing is that even with monotherapy, this resistance mechanism appears to play out. So ven on its own induces this, ibrutinib on its own induces this. The combination also induces this resistance mechanism. So it stands to reason that we're interested in this, and we're undergoing explorations to try to develop TLR9 and non-canonical NF-kappa B inhibitors. So another perhaps talk I'll come back and talk to you about on another occasion. So just to wrap up, a few final thoughts. I think it's fair to say that the treatment of CLL has come a very long way in the last 25 years. And I would argue, and I think most people would agree with me, that understanding the tumour biology has really accelerated the introduction of new and effective therapies. And it looks like BTK inhibitors and BCL2 antagonists are really set to transform the outlook for CLL patients. I didn't really touch on it today, but these drugs appear to even work in patients who are refractory to other forms of therapy. But at least for the majority of patients, they are not curative. Even the combo of ibrutinib venetoclax is not going to be curative for a significant proportion of CLL patients. And as I said, drug resistance is already starting to emerge. So the name of the game for us is to understand how this resistance comes about, understand how the bottlenecks that we put these tumour cells through, called therapy, is impacting upon the cellular response. And really it's the focus of our research team at BSMS. So we're developing in vitro models to try to mimic this cellular migration, because remember what ibrutinib does, it actually modulates that migratory potential. And we also want to design and test new drugs that block both migration and at the same time target the tumour cell in that lymphoid niche. And I've already alluded to two possible candidates there that we're advancing with. I'd like to end on a really optimistic note and say that in the next 5 to 10 years, I think it's entirely possible that the majority, if not all CLL patients, will be able to obtain a very, very significant and prolonged remission with therapies that we will tailor to their individual tumour. And it's absolutely possible that some may even be cured. And the reason why I'm confident about that is that I think we have indications that we are already curing some patients, rather fortuitously, perhaps even serendipitously, with the therapies we already have. So one more slide to go, one more data slide at least. So this is just to show you what we're developing at BSMS. So this is our in vitro circulating model of CLL. So this is the first really dynamic model of the disease, which allows us to mimic blood vessels, lay down endothelial cells and make really pseudo blood vessels that these CLL lymphocytes can traffic around in and then migrate through into a sort of lymphoid niche environment. So very exciting stuff. And what we've shown is that if we collect cells from the circulating compartment and the migrated compartment, what we call here the EVS, or the extravascular space, scientists all love an acronym, don't we? The field is absolutely awash with acronym. But what we can see is that these cells are very different in terms of their transcriptomes. So transcriptionally, they are doing very, very different things. And what's really interesting is that that appears to be modulated by microRNAs. So we haven't even discussed microRNAs today, but this is a small non-coding RNA species that actually regulates how RNA gets translated into protein. And what we can see here is that these cells that undergo migration in our circulating model, actually, they repress some of these microRNAs, which leads to the pro-survival BCL2 family of genes being switched on. So again, this story, I hope, fits together very nicely for you. What's really important is that this signature that we see from our in vitro model is replicated when we take lymph node biopsies from CLL patients and compare them with their peripheral blood. So the microRNA signatures that we are identifying in our in vitro model are actually very similar to those that we see in CLL, real CLL cells, if you like, from a patient ex vivo, from the two different microenvironments that we are interested in. Of course, because it's science, there are so many questions left unanswered. And actually, Andrea and I, we disagree about what we think the answer is here. But the question that we're asking is there something different about the cells that migrate versus those that stay in the circulation? Are they different genetic subpopulations? So, in other words, are they clonally distinct from one another? Or are they primed to migrate because of their phenotype, because they're transcriptionally different? And I won't tell you which one of those I think is the truth and which one Andrea is plumping for, but maybe someone might prize about me in the questions. Okay. What we have identified already, and this sort of validates what we've said to you about targeting the non-canonical signaling pathway, is that the miRNomes, the microRNA arrays that we generate, and the transcriptomics that we have point to non-canonical NF-kappa B activation because of an upregulation in a microRNA called miR322, which causes the downregulation of a regulator protein, an E3 ligase called TRAF3. And this leads to the stabilization of the master regulator of the non-canonical NF-kappa B pathway called NIC, NF-kappa B inducing kinase. Okay, enough of me already. Just remains for me to put up the most important slide to tell you that all research, all the best research anyway, is done in teams. And as you can see here, I have a long list of collaborators, all of whom are probably a lot cleverer than I'll ever be. So I am eternally grateful for these collaborations. The little team that we have at BSMS, led by Andrea and myself, and Simon has joined us recently. Really excited about what we're doing there. And the team on the far left-hand side here was my big team that we built up, Chris Fagan and I, in Cardiff. The final thing and final note of thanks is that we couldn't do any of this research without cancer research funding. And that, in large measure in the UK, comes from charitable giving rather than the government. So just to make it topical again, all the cancer research charities are suffering enormously because of COVID. So I hope that our government is going to recognise that fact and bury us in yet more debt in order to support these charities over at least the intervening period. Thank you for listening, and I'm very happy to take any questions. Okay.
So,
oh, we got 10 questions in the Q&A already.
Indeed. I think I'll just start off by saying thank you so much for giving up your time to talk to us on something which is truly, really, really inspirational. And I think, to quote you, the impetuousness of youth and arrogance was definitely not the theme which we got out of that. It was definitely an inspiring talk into the motivation behind your research into a really, really exciting area. So I'm sure it will have been appreciated by the audience. We do have quite a few questions, so I think we'll try and run through them as quickly as possible.
Fire away, Puru.
The first question, who was submitted by an anonymous attendee, which is a massive paragraph. If you're comfortable, can you please, can you please, whoever submitted this question, can you request to be made a panelist so that you can ask this on air? Because I think Professor Pepper may want to hear from you in a more interactive sense. We'll move on to Nagin's question, who is the other OHP co-president, and her question is, what is the life expectancy of a patient with CLL?
Okay. Hi, Nagin. Well, the answer to that question is very long and complicated, and you've already guessed from this evening's interactions that, you know, I'll never say anything in 10 words when 1,000 will do. But it's not an easy question to answer because CLL is so heterogeneous. So some patients with CLL have an incredibly aggressive form of the disease that I don't think should be characterized as a chronic disease at all. So their median life expectancy might be less than 2 years, which by any measure is not a chronic disease. Whereas I would say a good third of CLL patients will actually die with their disease, not of it. So one of the fascinations of CLL, and I hope if nothing else I've conveyed to you this evening what an amazingly interesting disease CLL is that, you know, we have this large group of patients whose disease down the microscope looks the same. So we have no clue morphologically who is who, and yet, you know, a large fraction of the patients will, you know, have a disease that will last their whole lifetime, the remainder of it, and it will not impact upon them health-wise significantly. So I'm sorry not to be able to answer the question really more accurately than that.
Thank you. I think the next question is quite interesting in terms of the advances in treatments which you mentioned. So this anonymous question is, what treatment options are available to patients through the NHS? Okay,
so all of the treatments that I talked about this evening are available through the NHS, by hook or by crook. So in the case of FCR, it's got NICE approval. And so, you know, all patients could be prescribed FCR. But as I said to you, and I hope, you know, you got the message, only a proportion of patients really do very well on FCR. And in fact, you know, we just had a paper bounced from Blood, which I'm about to turn back around, which identified that really it's about 14%, 14% of patients who have this long-term durable remission. That group of mutated CLL immunoglobulin genes with low CD49D expression and long telomeres. Those are the patients that will do really well on FCR. Everybody else probably shouldn't be treated with FCR. Now, you know, most patients should be able to get ibrutinib via the NHS. Venetoclax is a bit more troublesome, but there are clinical trials that you could be and should be recruited into if your clinician thinks that venetoclax is the right drug for you. So again, the short answer is all of the therapies are available through our amazing NHS.
Fantastic. So our next question is something which you said was probably going to be food for another thought. So I don't know if you want to go into too much detail, but why is CLL more prevalent in men?
Well, I mean, it's such an interesting question, Puru. Like I said to you, if you take out all of the obvious hormone-driven tumours, so breast, pancreas, ovarian, you know, there is an excess of cancer deaths and, in fact, incidence of cancer in the male population. And you wouldn't expect that to be the case, all things being equal. But we know, you know, that men and women aren't equal. Women are far superior to men. We know that. But, you know, all joking aside, I think the reality is, this is my spin, okay? I think that the female immune system is more competent than their male counterpart. And the reason for that is from an evolutionary biology perspective. You know, to be brutal about it, when a man has donated his sperm, his job is done in terms of passing on, you know, the genetics to the next generation. Whereas the female's work is only just beginning. The female's job is to nurture that, you know, well, actually to grow the fetus for 9 months to start with, but then let's be honest, you know, when does a human baby become independent? It takes a long time, right? And that's usually through, historically, from an evolutionary perspective, that would be the woman's role to achieve. And so I think from an evolutionary biology perspective, that makes sense, that the men don't need such a competent immune system, just from that perspective. But who can tell? I mean, there are smoking guns here. So women are more prone to autoimmune disease. And that is a signature of a primed immune system. So I think what I'm saying to you isn't without, you know, a rationale at least. But the truth is, it's really difficult to prove it.
Fantastic. Our next question is from our wonderful Director of Oncology, Emma, who asks, are the demographics of patients with mutated versus unmutated CLL different?
Are the demographics different? Well, what do you mean by demographics, Emma? So in terms of their characteristics, generally speaking, and this again sort of feeds into my narrative about male immune systems not being so competent. Generally speaking, the more aggressive forms of CLL, we have a higher proportion of males with unmutated immunoglobulin genes. But it's not a hugely or startling excess. In terms of other demographics, I can't think of any. I mean, generally speaking, again, this is another really interesting point. The more aggressive disease generally is seen in younger patients, and that certainly played out with my dad, for example. You know, he was diagnosed when he was 50, you know, so younger than I am today, and, you know, his disease progressed very rapidly. In fact, he was diagnosed in stage C disease. So, yeah, it fits. But there aren't any obvious demographic divergence, if you like, between mutated and unmutated cases.
Okay, so Emma, if you do want to specify further, do post again and I will pose it to Professor. Sure. Our next question is from our wonderful Director of Hematology, Nisal, which is, despite having CLL, some patients don't exhibit symptoms for years and some never need treatment. Does this depend on the CLL type?
Absolutely, it does, Nasal. Yeah, absolutely. So the vast majority of those patients with totally indolent CLL have mutated immunoglobulin genes. They have low levels of CD49D, and I've alluded to it a couple of times this evening already, but the strongest predictor by far for that indolent group is how long their telomeres are. So how long the bits of DNA, these very specialized structures at the end of linear chromosomes, are in their tumor B cells. It's fascinating. Now, I would argue, and again, I could give a whole lecture on just this for an hour, talking about how telomeres protect the genome and stop it from accumulating further genetic damage, which is the hallmark of human cancer. So as patients' disease progresses, we see this phenomenon called clonal evolution. The disease gets worse, if you like, gets more complicated. Yeah. So absolutely, we can identify those patients without a doubt. It's a really interesting sort of ethical, philosophical conundrum. If we can really identify those patients with unerring accuracy, should we even give them a diagnosis of a leukemia? Because let me tell you, the psychological burden of being labeled a cancer sufferer is enormous. I work with the Leukemia Care and the CLL Support Association in the UK, and I engage with patients, on a frequent basis, and they are all worried about their disease, and quite rightly so. They have this diagnosis that, in large numbers, they're told that they will watch and wait. The reality is that all patients watch and worry. And it has a massive detrimental impact on them psychologically. Not just them, their families too.
So if I may, Emma has specified in terms of the demographics. Would you like me to read the original question again? Or
Yeah.
Okay.
Well, just fire away. What did Emma say?
Whether it's younger or older patients.
Yep. Okay. So yes, generally speaking, it's not, the relationship is not a perfect straight line, but generally speaking, if you get your disease diagnosed at a young age, you have a more aggressive form of the disease. I guess that sort of makes sense, that if you're ill enough to require a visit to the doctor, or you're worried enough to think that your health might be not quite what it should be, and you're young, well, there's probably something wrong. And that's particularly true of men, because men don't generally go to the doctors very readily. If you look at all of the statistics around GP visits, women are much more likely to engage with their doctor, early than men. And that's a failing from men. It's not some hypochondria from women, far from it. Women are much more intuitive, I think, about their physical well-being than men. We tend to be deniers.
Just related to that strand, our wonderful director of research, Evie, has specified even further in terms of demographics. Can you see any other trends in terms of age, race, or socioeconomic status related to CLL within the research you've done?
I mean, socioeconomically, if you look at the globe, yes, for sure. And here's the rub: if you come from a poor country, your survival statistics will always look worse. And that's just a sad reality of advanced healthcare systems in the West outperforming, because we have access to all of the drugs, all the supportive care, that developing world countries do not have. In terms of race, there is a really interesting phenomena in CLL. So that actually, as as a proportion of, you know, different ethnic groups, CLL is more prevalent in Afro-Caribbean communities, which is very, very interesting. Yeah.
Fantastic. Our next question is from Christabel, who says, you said BTK and BCL2 are important targets for CLL and that drug resistance is a big issue. Does that imply that BTK and BCL2 are adapting to treatment, or that other genes may be contributing / amplifying the CLL? And if so, has there been any progress in identifying any other target genes that can contribute to CLL?
So, really, really interesting question. And as usual, I have a very long answer. But let's try and break it down. Okay, so some of the resistance that we see to both venetoclax and BTK is entirely rational. So we can see the development or the growth, the clonal growth or clonal expansion of cells that have mutations in BTK itself, yeah, the gene BTK. So it makes so it's a non-synonymous mutation that changes an amino acid in the protein that allows ibrutinib not to bind the protein as well. Okay, so the on time for ibrutinib is diminished in those cells, making the drug less effective. So that's entirely rational. And we also see mutations in a downstream signaling molecule called phospholipase C gamma. So again, incredibly rational that even if you get the on effect with BTK, the downstream signal for phospholipase C gamma is switched on by a mutation in phospholipase C gamma. So if you like, the tumour is bypassing the BTK block. So that's very rational. And if we look at BCL2, the same is true. We see mutations occurring in the BCL2 gene itself, which stops or diminishes the binding capacity of venetoclax to BCL2. So one of the joys of working on these new targeted therapies is we sort of can predict where the resistance might come from. Now, infuriatingly, but actually really intriguingly, only a fraction of the patients that get resistance to BTK inhibitors or BCL2 inhibitors actually carry one of those overt mutations in those pathways. So clearly, there are other genes, other pathways that are being invoked in the remaining tumour cells or resistant tumour cells. Now the question is that selection of a subpopulation of cells that already have those pathways overactivated, or is it induction? Has the drug, exposure to the drug, forced those cells to change? And if there is an archetypal question in cancer cell biology, it is that one about induction versus selection. All I will say to you on that question, because I don't know the answer, is that rather encouragingly, I think we've seen, with the advent of next generation sequencing and deep genetic profiling, we've seen that the selection effect is very, very obvious in the context of therapy. And the reason why I think that's encouraging is that I always imagined in my younger, more impetuous youth that we would never cure cancer because cancer would always evade us. It would always be like chasing shadows, that we would see this evolution of the tumour. And in fact, if you look at the next-gen sequencing data, most of the problems that we encounter later with therapy could have been predicted by looking at the early analysis, the sample pre-treatment, in deep enough detail. So what I'm saying to you is that I think we will crack this nut, in that if we're smart and we can work out what all these mutations mean and what influence they may have in the context of therapeutic exposure, we should be able to design strategies to overcome them eventually. But it will require large numbers, probably, of these targeted inhibitors given simultaneously or serially in order to achieve that goal. Does that answer the question?
I think so.
If not, I filled 5 minutes of time anyway.
Just on that tack, we do have 5 minutes left. I will leave it up to you if you want to carry on answering the remainder of these questions.
Keep going. Fill your boots.
Okay, so I think it'd be quite nice to go to a question from a fresher.
Great.
If CLL is poorly or untreated, can it change into ALL?
No.
Okay.
There you go. That's my shortest answer of the night. But no, in all seriousness, no, it cannot. Now, what is really fascinating, so I gave a one-word answer just to prove that I can, but now I'm going to give my usual long-winded answer. So if you look at gene expression profiling or transcriptomic profiling, if you do non-supervised or unsupervised hierarchical clustering, in other words, if you look at pattern recognition in the gene expression profiles from loads of different tumours, CLL and ALL sit right side by side. So they get clustered together. Now, intuitively, that should make sense to you all, because what's the cell of origin of ALL in the vast majority of cases? Puru, you've got to answer because you're the only one who's got a microphone.
Cell of origin, has it?
Yeah. So
it must be of lymphoid origin.
Yeah, acute lymphoblastic leukemia. Lympho, yeah, speaks to the lymphoid origin. What are the two cell types in the lymphoid, the large cell sort of bifurcation in the lymphoid lineage?
T cells and B cells.
Perfect. So are most ALL patients B or T?
Module 102.
Go earlier in the alphabet.
B cells?
B. Most CLL patients are BCLL. So it stands to reason if the cell of origin is the same, they're going to cluster the same. Because remember what I said to you right on that, I think the first slide, right? That the epigenetic program of a B cell is pretty much fixed, you know, so all B cells have this genetic picture, this blueprint stamped on them, which regulates the genes that make them a B cell, yeah? So it stands to reason that BALL and BCLL cluster together. The genes that regulate their behavior are quite commonly shared, but you cannot revert from CLL to ALL. But what is really interesting, of course, is that some CLL patients have a very aggressive disease that is, in pathology terms, in terms of disease progression, is more like ALL than CLL. Yeah.
So I think we will move on to a question from Evie, and I don't think it's as related to your talk, but very much interesting considering the theme, which was a very personal connection you have to CLL, which was your driving motivation. So I'm sure there's quite a few medical students out there who may be doing research in their future careers, potentially even educating. So what do you, are there any words of advice you have for them in terms of how to get involved with research, whether they should do it as early as possible as they can in medical school, and sort of progress that into their careers side by side alongside their clinical work as well?
I mean, the first thing to say is I think without clinical academic researchers, so clinically qualified people who are curious about the biology of disease, we will never really significantly advance. That is absolutely crystal clear. Bods like me need clinical academic researchers to work with, you know, because without the people who are seeing the patients and understanding what their needs are, you know, I need that in order to A, serve as a motivating factor, but B, to understand what's important in the disease, you know, so that we can focus our research attention on what patients need and what patients and their families want. So I would absolutely encourage all of you to get involved in research as much as you possibly can, you know, and that can take all sorts of guises. You know, it doesn't have to be, you know, putting on a lab coat and holding a pipette. You know, you can get involved in doing audit, for example. I mean, it sounds, even the name sounds boring, doesn't it? But actually, clinical audit can be really interesting and insightful. And often, clinical audits throw up very unexpected things in clinical practice that sometimes lead to clinical practice change, which is really important. So, you know, that's what healthcare improvement research is all about, really. Understanding what we do and why we do it and whether we could do something better. Yeah. So my advice is try and seek out opportunities. I mean, it's so difficult. I mean, we're in a COVID situation at the moment. Getting into labs is going to be nigh on impossible for the foreseeable future, I would think, because, you know, my full-time research team can barely get in the lab, let alone, you know, give opportunities to students to get in the lab, which is such a shame. But, you know, if you're innovative enough, if you're, you know, ingenious enough and actually determined enough, you'll find a way.
I suppose I have a question of my own from looking at the history of one of the drugs you were talking about. Do correct me if I'm wrong. I think it was the clinical trials for ibrutinib, the graph which you displayed,
Yep.
and that spike in lymphocytosis within the 50-day period. During that trial, would that have been classed as an adverse effect, and would it have caused it to be sort of shut down? And in that case, if the ethics board did get involved and stop it, how did they manage to carry on to get the data and find out that this was just a blip and actually there was a therapeutic effect?
Yeah, so all clinical trials, I'm sure you know this, Puru, but you know, all clinical trials have these very necessary governing committees called data monitoring committees. And they look at, you know, the effect or impact of a new experimental drug on a patient. And they are really, you know, the final arbiter of, you know, good taste, if you like, but, you know, in this context of determining whether that trial should stop or carry on. So they're constantly balancing, you know, the risk and the benefit to the patient. So they are really the patient's advocate supreme. Now, bear in mind that the doctors that are giving the drugs to these patients are also their advocates. I mean, I have almost, well, so rarely come across a sort of narcissistic doctor. They do exist, believe me. I've met a couple, but they are rare breed. You know, most doctors care passionately about their patients and only want what's best for them. And those are the types of doctors that we should be training in BSMS. And in large measure, I'm sure that is true. But yeah, I mean, going back to the ibrutinib story, like I said to you, it was a squeaky bum moment. I really do, you know, I think that's true. The doctors that gave those first doses of the drug must have thought, in fact, I know that they thought, you know, crikey Moses, what have we done here? We've caused this disease to grow out of control. But fortunately, ibrutinib was given in an era where, you know, we had imaging technology available to us. And so they could very quickly determined that actually it was having a significant impact on lymphoid tissue. And so they put the two and two together and realized actually this completely unexpected effect had occurred, that the tumour was being forced out of the tissue into the peripheral blood, and that was giving the spike in lymphocytosis. But you're right, it could have gone either way. I mean, the trial may have been stopped prematurely, and then we wouldn't have this wonder drug that we now rely so heavily upon, actually.
Fantastic. Thank you very much.
You're welcome.
I think I will summarize this question because I think it's important and the person doesn't want to come live on air. And it's talking about the clinical trials and the ability to build up tumor banks, which can be used for research for decades after the trial is completed. But there may be a concern that this can be owned by pharma companies and exploited and sold them to other companies for profit. Do you think this is a necessary trade-off for research? Or do you think there are other ways we can collect material and still accomplish the same level of research output?
Yeah, I mean, it's a vexing one. And, you know, I didn't today, but I have in the past sort of played slightly devil's advocate and talked about pharma in slightly less than glowing terms, but that actually wouldn't be fair. Pharma companies are first and foremost businesses, and we need to remember that at all costs. You know, their mission statements are always full of altruistic words, but in fact, they're there to serve their shareholders. And you need to remember that. So what we get from them should always be viewed through that prism. But they should never be characterized as the devil incarnate either. You know, they do remarkable things. They bring new drugs to the market that we would never bring forward if we were left to our own devices. They invest billions and billions of dollars, pounds, into the drug development pathway. So we need to play with pharma. We need to work with pharma and get what we need from pharma in order to bring about positive healthcare change. But what I would say to you about biobanking is that big pharma seldom own the biobanks that we generate through clinical trials. So they know that it's bad press for them to take patient material, you know, from a clinical trial and store it at Roche Diagnostics or wherever, you know. So they don't. They generally pay for the biobanking to be done by a clinical trials group. Even if it's a drug company sponsored trial where they're providing the drug for free and maybe quite a lot of the supportive nursing care, they will usually invest that tissue in an academic centre. And throughout my career, I've benefited from access to those biobanks of samples. You know, and Andrew and I are still benefiting from those samples, you know, and bringing forward our research, which is not drug company sponsored at all, you know. So I think we needn't worry too much. We need to be vigilant. We need to make sure that the status quo stays like that and that patients really are donating samples that will be there for posterity and will be used to advance medicine in the way that the patient who donated them envisaged. But, you know, selling them to big pharma is not something that really happens very often. A lot of people have actually tried and failed miserably, thankfully, to make businesses out of biobanking. You know, it doesn't work. It costs an awful lot of money to keep cells banked down in a really good state so that they can be used again to do biological research or genetic research. So again, I've waffled on, but essentially, I'm a massive fan of biobanking. I think we should take pharma for what they can give us, but keep in mind what the ultimate aim of pharma is.
I think Mo would like to come on air. I don't know if we've asked your question, but Mo, you should be allowed to speak now. And if you do have
you actually just answered, you asked my question already. It was the one about the biobanking.
Fantastic. Okay, thank you.
Well, I hope I answered it properly for you, Mo.
I think we'll move on to our next question, which is from Abby, and that is, are follicular lymphoma and CLL similar cancers?
Similar. So they're B-cell malignancies. And what is really interesting is that follicular lymphoma is the archetypal BCL2 tumour. So follicular lymphoma is characterized, almost all patients with a diagnosis of follicular lymphoma have a particular translocation event called a T1418 translocation event, which for the geeks amongst you, and even if there aren't any amongst you, I'm one, so I'll just tell you, what that does is it brings the BCL2 gene under the control of the immunoglobulin heavy chain gene promoter. Now, the immunoglobulin heavy chain gene is an incredibly powerful promoter, so it drives the expression of BCL2. Now, what you would imagine, wouldn't you, is that follicular lymphoma would therefore be the archetypal tumour suited to treatment with venetoclax. It just isn't. And we do not know why. We cannot understand why follicular lymphoma patients do not respond well to venetoclax. But the response rates are pitiful. I mean, it's like 25% response rate, where you'd expect 100% response. And we just don't know why. So more research needs to be done. But yeah, follicular lymphoma and CLL are sort of cousins, I would say. That's the way I would describe it in disease terms.
Would you like to wrap up with one more question and call it a day?
Fire away.
So this is from Ellie, who's our wonderful phase 3 rep and really got the society going. So just a shout out to her. And her question is, do patients die from serious infections? And I think in the climate we're in, it's probably a very pertinent question.
Yep.
Due to their immunosuppression on these therapies? And if so, how do we protect them?
I mean, I think this is so pertinent to the current situation we're in, Ellie. You know, I think shielding is the most effective way of protecting CLL patients at the moment. I mean, if you look at the global data, it looks like there are an excess of deaths in CLL patient communities with COVID. So if they get the disease, they end up being more frequently hospitalized and dying of COVID infection complication. It's still not enormous numbers. So the data, you know, we need to be cautious about the stats because it could just be a sampling error. But it looks to me like, on balance, CLL patients are more vulnerable to COVID than the background population. Of course, what we need to do is normalize that against age, because we know that age is a risk factor in COVID as well. And that, to my knowledge, has not been done yet to any great extent. But yeah, the safest thing we can do is to keep our vulnerable in society, you know, isolated as much as we possibly can. Now, I understand that that's tricky and actually not desirable for them as well as for us, because it means that they sort of, I mean, shielding is a really nice word, isn't it? It sounds like it's a very protective, nurturing sort of environment, but actually the reality for a lot of patients is that they're left alone. And that's horrid. Human beings need human contact. We're pack animals, aren't we? We like to be near each other. So yeah, my take-home message is that, you know, we need to be still shielding these patients that are vulnerable, because I think there's a chance that, you know, they are, at least a significant chance that they're more vulnerable to COVID. Yeah.
I think that's a very, very nice point to end on, I think, in terms of the current climate we're in. If it's okay, the remaining 7 questions, if I email these to you, would you be able to sort of just pen down an answer for them, and then we'll publish them on our social media?
For sure.
I'm conscious we have already eaten up plenty of your evening. And all that's really left for me to say is this has been fantastic, but there is a tinge of sadness for me because I'm just envisaging what it would have been like in person. And I'm sure one day we will be able to invite you back into that Cowan Lecture Theatre again with a full audience, and we'll be able to interact in person. And I'm sure it will be, if not better than what we've had this evening. So my genuine thanks to you for giving up your time and accepting our invitation to come and speak.
It's an absolute pleasure, Puru. Honestly, it really is. I've loved it. I talked about my favourite subject. I talked about my favourite man, my dad. You know, so what's better than that in life? You know, so and I hope that I conveyed a little bit of something of my passion, at least, for my subject, and hopefully a little bit of insight and knowledge as well. Just a little, anyway. But as for meeting in person, whenever we can, I will be there. And I would love to meet you and your committee and everybody else that wants to come along. Yeah, it would be fantastic.
I look forward to that day with great earnest, I think. But a couple of housekeeping notes, I think, for the rest of the society, if you are interested. On the 13th of October, we'll be launching our book club, where we'll be talking about the book Being Mortal by Atul Gawande. The 26th of October, we have Dr. Norris Igbenoweka, who will be talking about sickle cell disease in the context of Black Lives Matter. So that will be a great event if you can attend. The registration link is in our social media, so please do sign up. And then we're full steam ahead. We're not going to stop. On the 9th of November, we have a series of how-to research talks, which will be starting. And the link for that will be in our social media too. I've also been asked to give a shout out to the Tea with Tarina event, which is happening at 8 o'clock, so just a couple of minutes after the end of this. So if you're a fresher and you haven't had your evening sort of enthralled in society events, please do go and attend that. And I think I'll just close by thanking you, Professor Pepper, once again. Absolutely fantastic. And you've already sort of left us on a cliffhanger with almost 4 different other talks, which you've proposed.
Can you bear it? That's the question, Puru.
Most certainly, and I think the struggle will be to try and fit them in within my tenure as co-president with Nagin, because I want to be there for every single one of them. But yes, thank you so much. Thank you for all the attendees who've remained. I think at one point we were at 75 and 60 have still remained. So thank you so much. Have a good evening and stay safe. So
fantastic.
Thank you very much.
Good night, everybody.
Take care.
Bye.